we're going to go through a very interesting area of the study design that's all about chromosomes, which is something that we've actually talked about quite a bit already. And chromosomes are very important to this guy and very important to you and me. But this guy, he's lucky. He was born in an extra chromosome, and a wise person once told me that people like this guy who has Down syndrome are actually the most amazing people in the world. All right, so pause the video here and have a go at the 3 quick questions, and we'll go through them together in class. All right, so big chunk of the study design you're going through. All right, we want to know about the role of chromosomes as structures. We want to know about the role of chromosomes as structures that package DNA, their variability in terms of size and the number of genes they carry in different organisms, the distinction between what we call the autosome and the sex chromosome, and the nature of our favorite word in the world, homologous pair of chromosomes, because we have one maternal and one paternal chromosome carrying the same gene loci. Very important term. This is all stuff that we've covered in area of study 1 a little bit. We're going to go through in more depth, but it's all stuff also from year 10. Okay. We're also going to look at the presentation of an organism's set of chromosomes as a karyotype that can be used to identify some chromosomal abnormalities such as Downs, Klinefelter's, and Turner syndrome in humans. All right, so question for you, what's happening here in this picture here? What is happening? We can see that we have cells that have been radioactively stained. Their spindles have been radioactively stained green, and their nuclei, and especially the structures inside the nuclei, have been radioactively stained red. What are the red stained parts? Hopefully you can identify that they're the chromosomes. And why are they moving? Because the cells are dividing. That's mitosis. There's a very cool representation of mitosis there, and then mitosis over here. Not representation, happening in real life, actually. All right, so we have to know about 2 different kinds of chromosomes. The first kind of chromosome is the prokaryotic, the simple, okay, the basic, the first kind of chromosome. Prokaryotic chromosome is circular, okay? The second kind of chromosome you have to know about is eukaryotic chromosome, and eukaryotic chromosome is linear. Okay, that's all you have to know about prokaryotic chromosomes in year 11. We are going to talk about it a little bit in terms of the pigloprac, but that's all you really have to know about them. All right. Okay. Let's talk about the role of chromosomes. And the role of a chromosome, it's to protect your DNA. Okay, it's going to take your DNA, store it really nicely and packed, and protect it. Okay, much like a library actually provides the way to store and organize books, a chromosome provides the storage and organization for our DNA. Okay, but also like a library, if we look at this library, yeah, sure, the central feature of these books, most of a library is actually made up of structure. Okay, there's the ceiling in the library, there's like the furniture in the library, there's the structures in the cabinets that hold up the books. As much as the books are the important part, most of the library isn't made up of books. Okay, and that's the same with chromosomes. Okay, chromosomes are mostly made up of what we call histone proteins. Histone proteins, which is a type of protein that its function is literally to package up the chromosomes, okay? So obviously we find our chromosomes packaged up in the nucleus. Usually they don't look like that. Usually they look like this, which we call chromatin. Okay, but chromatin works much the same, okay? And if we look at a chromosome, what a chromosome is a coiling and then a supercoiling into what we call structures called nucleosomes. And what a nucleosome is histones, which is this protein, plus the DNA. Okay, and if we actually unravel these nucleosomes, we can pop out the histone proteins, and we get DNA double helix. All right, this is just another diagram showing how chromosomes are packaged into this DNA. If you follow this link on the PowerPoint, you can actually see this in not real life, it's a representation, but it's a biochemically accurate representation that's actually really, really interesting to see. And I think it's really cool because it's a representation that's actually used worldwide, but it was made just down the road in Melbourne. All right, so what chromosomes look like, okay? Chromosomes, we often represent them like this. We represent them duplicated. And yeah, sure, this is one chromosome, all right? This is one chromatid, and it's a chromatid, okay? But actually, most of the time, chromosomes actually look like this, right? Just a linear, unduplicated chromosome. All right, let's break down the structure of a chromosome more. We're going to often represent it as this X just because it's the more common way that we focus. The reason why is because the best time to see chromosomes is actually during meiosis or mitosis, and that's the chromosome. It looks like this X shape, and so we often represent it like this. Okay, so chromosomes have two identical chromatids. Okay, so each one of these is identical to the other. Okay. And each actually contains one whole DNA molecule. So if we actually unravel the DNA, it's all one long molecule of DNA. Okay, one on one side, one on the other side. At the very middle, we have what we call the centromere, and that's the joining point of the two chromatids. Okay, and we have two different arms to our chromosomes. Okay, it's not about the top arm or the bottom arm. It's actually about the size. Okay, so the P arm is the shorter arm. So in this case here, the top arm is the P arm, and the Q arm is the longer arm. So in this case here, the bottom arm is the Q arm. And then chromosomes at the very, very end, they have this very important structure called telomeres. And we've telomeres a little bit, and telomeres are very important because they protect the ends of the chromosomes. They're almost like end caps, okay? And each round of mitosis in our cells, okay, when our cells are duplicating itself, renewing themselves or something, those telomeres actually degrade. So the DNA has to replicate and the telomeres degrade. And that's why we can actually say telomeres are a good indication of life expectancy. Which is pretty amazing. So there's quite a bit of variability in chromosomes, and although all eukaryotic organisms have the same structure, chromosomes actually vary quite a bit both in size and in number of genes that they actually carry. So let's first look at human chromosomes. Chromosomes. And in these examples, we're only talking about what we call sex chromosomes. And in human chromosomes, females actually have two versions of the X chromosome, and they are genetically identical in terms of the type of genes that they carry, and they're genetically identical in terms of their size. Males, on the other hand, they have non-homologous sex chromosomes where we have the X version of the chromosome, but the Y version is simply a shortened chromosome. Okay? But that's not the case in terms of all animals. For example, flies have the exact same as humans in terms of having X or XY, but in flies, simply the female has two X chromosomes that are the same. And then for the males, the X chromosome and the Y chromosome aren't different sizes, but they just have completely different genes on them. So they're not not homologous. And then we can even have examples of things like worms, where a hermaphrodite worm, okay, there's no actual female worms, okay? They're either a male or they're both. But a hermaphrodite worm just has two versions of that X chromosome, and then a male worm only has one version, and then the other one is just missing. There's none there. That's why we call it a zero, okay? And they're just different ways that we can vary these chromosomes. So just like we've been talking about, every chromosome has a pair. Okay, so in diploid 2N or eukaryotic organism, each chromosome has that homologous pair, which just the definition of a homologous pair is that it's the same size and it has the same genes at the same gene loci. But remember, possibly different alleles to those genes. Okay, and we call each homologous pair the paternal and the maternal copy. And that just means paternal means from the dad, maternal means from your mum. Okay? So here's homologous pair chromosome 1 and homologous pair chromosome 2. All right, now let's talk about karyotypes. Okay, karyotypes are very interesting because a karyotype is the entire set of chromosomes of an organism, and they can be visualized by freezing a cell which is just about to divide and staining their chromosomes that we have a series of bands appearing, okay? And chromosomes are then organized into what we call the autosomes, which are your normal chromosomes, and your sex chromosomes, which are the chromosomes that determine whether you're male or female. Here we have an example of a human karyotype, okay? And if you can notice here, each chromosome, so they're ordered by size, okay, and then we number them by size as well. So chromosome number 1 is just simply the biggest chromosome that humans have, and then it goes all the way to chromosome number 22. So from 1 to 22, we're talking about autosomes, right? And then finally at the end, we have these guys here. And then, so if you're a female, you're going to have XX, and if you're a male, you're going to be XY. And so XX and XY are our sex chromosomes. Okay? Notice that each chromosome has what we call these banding patterns, okay? And as much as it's a bit hard to tell, each homologous pair is actually genetically identical in terms of these banding patterns. So their DNA is similar enough that they have these same bands, okay? And the bands, by the way, are just dictated by the number of A and T's in some regions and the number of C's and G's in other regions. So one of these bands one way and the other one bands the other way, okay? So now let's look at some abnormalities in terms of number of chromosomes. The first abnormality we're going to talk about, you certainly have always have heard before, okay? And that's Down's syndrome, okay? And Down syndrome presents like this, okay? And it's characterized by 3 chromosomes number 21, and that's what we call a trisomy 21. So tri meaning 3 and somy from the word chromosome. So 3 chromosome 21s, okay? Another very common abnormality in terms of number of chromosomes is what we call Turner's syndrome, okay? And Turner's syndrome is actually characterized by 1 less chromosome, and specifically no copy of chromosome Y, okay? People with Turner syndrome, such as this actress from the TV show NCIS, present as female, okay? Generally, they present and identify as female, but we say that they have this XO karyotype, okay? And that's simply because there's nothing at sex chromosome position 2, okay? But because they don't have the Y chromosome, they don't develop the male features. The last kind of um, type of abnormalities in the number of chromosomes is what we call Klinefelter syndrome. And Klinefelter syndrome is what we call the XXY karyotype, or it can even present as XXXY karyotype, but XXY is certainly a lot more common, so don't stress about that other one. And as you can see here, this individual has both the double X chromosome and the Y. So now the question is, what's their sex? Yeah? And this actually raises for us a bunch of biological facts, which are things like Turner's syndrome and Klinefelter syndrome, and starts hopefully making us question our societal norms. Okay, let's look at why this happens. Okay, and the reason why it happens is a process that's called non-disjunction. Okay, non-disjunction isn't a normal process. It is a abnormality, and it's usually not good. Okay, and if it happens in your sex chromosomes, the results are actually not that ridiculously crazy, okay? Turner syndrome and Klinefelter syndrome individuals can live completely normal lives. But if it does happen to your autosomes, especially your big number one, number two, number three autosomes, it usually does lead to a miscarriage, okay? And simply what happens is when your cell goes through meiosis, this is meiosis here, okay? Um, in meiosis 2, so this is meiosis 2 there, instead of your sister chromatid splitting, like here, as you can see, normal sister chromatid splitting, that doesn't happen, which means that we're left with a gamete with an extra bit of chromosome, okay? And that actually raises the question for us to consider something, which is this. This is Caroline Cozy, okay? And Caroline Cozy is actually a person that was born with Klinefelter syndrome. They are X, she is XXY. So she has both the female and the male chromosome, but she identifies completely as a female. She actually was a British supermodel. She was in one of the James Bond movies, and for a long, long time, she had a highly successful career. But unfortunately, due to her being XXY and this was ousted, right? She actually fell from fame and lost her opportunities in modeling, and it's a very sad story in that case.